Check, check. All right, homeostasis. Your study guide, not homeostasis, hemostasis. Two different things. I'm telling you basically this is going to be a short answer question on your assessment. And by the way, if there are some of you who are planning on taking this before you leave Wednesday, right, which would mean you want to take it tomorrow, unless you're taking it today, or you want to arrange something early on Wednesday morning, I don't know what time you guys leave. No. So tomorrow would be acceptable. You need to come see me, please. I need to know what the plan is, because I've not heard it from anybody yet. All right, so again, there are three big pieces involved here with this hemostasis. And again, hemostasis is just simply, we don't want you to bleed to death, right? When you start bleeding, we want it to stop. And there are three main pieces that are involved in this. Now, a question was asked just a couple minutes ago, do I need to know the whole litany of all the things that are involved? You don't. And again, your textbook gives you a decent list, listing everything out. It's way more complicated than that. I'm talking about like hundreds of steps more complicated than that. I don't need you to know all of that. I do want you to be comfortable with these three main methods that it uses: blood vessel spasms, platelet plug formation, and blood coagulation. Let's talk briefly about all three of those pieces. Remember, the blood vessels, especially the arteries, oops, it's not in this slide, it's in the other one, they have the little muscles around them, right, that allow them to contract when you get cold, right, and to open when you get hot, right? So when a cut occurs, it reflexively makes these blood vessels contract. And again, we call that a vasospasm. Vaso meaning vessel, spasm to tighten, right? So it's purposefully, when it's cut, it will tighten to almost seal itself off. It's actually fairly clever. It's going to like purposely prevent the, again, think of a hose that you've just cut off the end of it. Water is just going to come pouring out, right? Well, imagine if the hose just like self-clamped. That's what your blood vessel's doing. Pretty impressive. So that's just slowing that outflow of the blood. It only lasts a couple of minutes, but it's enough for the next pieces of the puzzle to take place. The platelet plug formation. Remember platelets in the blood, right? They're like little pieces of cells. And what they do is they clump together. They like stick together. And that starts to form like just a little blockage in that broken vessel. Because remember, now it's gotten smaller, right? It's purposely constricted. So that helps to get that initial stoppage of blood flow. Sir?
When they stick together, do they stick together like a spring like that?
Reasonably quick, yeah. Yeah, certainly. Because again, your body's trying to prevent the blood flow from just blasting all over the place. So yeah, it is reasonably quick. And they call that a platelet plug. This basic little blob, little clot, if you will. It's not exactly clotting. They're two different things. Your textbook has a nice little yellow box that talks that there's a difference between blood clotting and this agglutinizing they're talking about.
So these are all steps that like work together. It's not like
Exactly. Exactly. It's kind of the three main things that help you stop bleeding. Yep. And the last piece that really is the most detailed, I can't get this thing to go here. There we go. Sorry, this is showing that platelet plug being formed, right? The platelets are in the blood all the time. When there's a cut that happens, the platelets start adhering to each other as well as to the ends of the broken vessel, as well as exposed collagen that's present there, and that makes that platelet plug to help control the blood loss. Again, the main or the most effective way of hemostasis is this blood coagulation. And this is the piece that has, again, the multiple steps with all the different clotting factors and all the different enzymes. And it's an amazing process. Again, I said this to you before, but there are some folks who study origins who say there's no way this could have happened on a step-by-step, gradually incremental basis. You had to have the whole system in place, otherwise you would have bled to death with a simple paper cut. So, very complex, very complex. Here is the key. The major event in blood clot formation is taking fibrinogen, which is soluble, right? Just dissolve, that's one of the proteins that's in your blood, right? And changing that fibrinogen into insoluble fibrin. So the same protein is changed when this blood clotting needs to occur. And it goes from being something that's just dissolved in the blood, no problem, to something that will not dissolve in the blood. And that will then start this, again, along with the other pieces, the vasoconstriction, the platelet plug, will start to make that blood clot, to actually keep the blood from simply flowing. Does that make sense? So again, that would be the main piece there. They talk about other pieces, again, the prothrombin activator, converting prothrombin into thrombin. These are all good things. Again, I'm looking for a brief description about these three pieces. The blood vessel spasms, the platelet clotting, and then the blood coagulation. If you have not, I would encourage you, check out, there's a nice outline at the end of each chapter. I'm sure you know this. That does a very good job of just giving you briefness. Here's what this chapter talks about in fairly brief detail. A good place to spend some time. Any questions here? Sure.
Fibrogen is just like a generic term, or is that like a specific?
No, that's a specific protein that's dissolved in the blood. Yeah. Mm-hmm. And of course, the whole point, of what makes this system so amazing is we want clotting to occur only in places where there's a cut. If the clotting occurs other places, well, then you have significant problems, right? And again, they talk about a blood clot that forms abnormally in a vessel. We call that a thrombus. If it dislodges and now it's literally moving through your blood system, we call that an embolus. Both of those are bad things, right? A blood clot in a place where there is no cut, this is a problem. Are you with me there? Please. In other words, if you think of so let's say in your leg, for example, you're sitting on a plane. This is a very common thing that happens. You're not moving, right? The change in pressure can actually cause blood clots to form. And you get a blood clot that starts forming in your calf. This is a very common place for this happening with older folk. As long as it stays in your calf where it formed, that's what we call a thrombus, a deep vein thrombus, a DVT. Maybe you've heard of this before. That blood clot, if it breaks off, now it's just floating in the blood, and as soon as it comes to a vessel small enough, it'll lodge there. That's what we call an embolus. So what can happen is you can have a blood clot in your calf, a blood thrombus, that then breaks off and travels and lands, let's say, in your lungs. Now that's what we call a pulmonary embolus. That cuts off the blood supply to one of your lungs, the lung tissue can die, and therefore you can die. Does that make sense? If that same thrombus goes and lodges in your brain, that's a stroke, right? It's cutting off blood supply to one part of your brain. Again, a bad thing. Does that make sense? These are not on your study guide, so I'm not going to ask you the pieces, but that is the difference between them. There was a hand that was up. Maybe I answered it. Yes. All right, let's keep going. Blood transfusion, blood groups, and transfusions. So this is all based upon what we call antigens and antibodies. And there's a very nice picture in your textbook that kind of talks and kind of shows the differences between. This is over in chapter 12, where I am here. That shows it there on page 344, 345. There's good pieces there that show that. So an antigen are little proteins that are on the surface of the red blood cells. So remember our red blood cells that are carrying the oxygen? They have these little proteins on the outside of them. And your body also has antibodies that will basically attack those antigens. Does that make sense? That's how antibodies work, right? They're specific to a certain protein. If I had an anti-HANA antibody and it sees HANA, it's going to attack, right? And it's the interaction between these two things that determines whether or not different blood types you can accept. It used to be back in the day, we would transfuse, if Sonali was not well, I could give her some of my blood. And maybe it would be okay, but maybe she would die because the blood would just start clumping together, which, clumpy blood doesn't really work too well. And they couldn't figure out quite why. They said, well, there must be some reason that sometimes I can give her blood and it's fine, but if Ryan comes and gives her blood, it doesn't work. The blood looks the same, but there must be something different. Does that make sense? And that's the interaction between these antibodies and antigens. Your question? So the antigens are actually on the red blood cells themselves. The antibodies are the immune system's response to it. Not necessarily. It's just that when we're thinking about it like in a bacteria or viral kind of thing, yes, it's bad. But these are just present on the red blood cells. There's nothing you can do about them. They're just part of the structure of that red blood cell. And the body just then reacts to them. And these antibodies aren't necessarily because you've been exposed to something else. They're just present there in the plasma. They are. Mm-hmm. Yes. Right. We'll determine how they react together. Right? Normally, if you're not exposed, pop this up here so you can see it. You've got basically 4 types of blood. You've got A, you've got B, you've got AB, and you've got O. So type A, they have A antigens on the red blood cells. Yes, ma'am? We'll get to the RH in a moment. This is kind of a separate. The ABO is a separate kind of blood type, and then the RH positive or negative is a different. They're different pieces. So, like in other words, you will be A positive or A negative or B positive. Does that make sense? You label both of them. So the A blood has A antigens and anti-B antibodies. So on the red blood cells, it has these A little proteins, these A little antigens. And in the plasma, it's got antibodies to attack any B proteins, any B antigens. Does that make sense? B is just the opposite. B antigens, A antibodies. Type AB has both, on the red blood cells, both of those A and B antigens, right? The proteins that are on the red blood cells, it has both of those. And no antibodies, which makes sense, right? Otherwise, it'd be attacking itself. So there can't be any antibodies for AB blood. And type O blood has neither antigen on the red blood cell, but it has both types of antibodies, which again makes sense. If it has 2 antibodies for both A and B, you can't have anything on the red blood cells, otherwise, again, it would clump together. We're good so far? Yes. Right. Because if O got A or B, the antibodies that are there, the A would attack and they would clump. Yes, ma'am.
So does that mean type AB blood they get sick more?
No, it's just rarer. And they have to make sure that they get a very specific kind of blood, right? They can't have any antibodies in the blood. So can they get A? They can't get A because A has B antibodies, right? They can't get B blood because it has A antibodies. They can get AB or they can get O. That's really their only possibility.
So how do people receive O blood if it has, because isn't that the universal donor one?
Mm-hmm.
So then how do they receive if a B person receives it? They have A antibodies, but then the O has B antibodies. So wouldn't that fight each other?
The O has got neither antigen on the red blood cells. So even if you have the antibodies, there's nothing on the red blood cells to attack.
Well, wouldn't the antibodies on the O blood attack the ones on the B?
They would just give you the red blood cells. They wouldn't give you the plasma. Yes, very good. Yes. Because the antibodies are in the plasma.
Is the antibodies in the plasma for the other types?
No, all of them. All of them, yes. Again, this is why when they collect your whole blood, they will actually take it and then siphon out the plasma and the red blood cells. Again, unless it's a specific kind that everybody can get. Does this make sense, my friends? Yes. Well, it's important to know that there are these interactions between the different types, and you have to get the right type, otherwise you'll die. Right, with a blood transfusion, exactly. Quickly, let's roll to the next piece, RH, positive and negative. That basically there is a protein that's on the surface of these red blood cells that, again, they're labeling it as Rh positive or Rh negative. Are you with me? If you have this Rh protein present, you're positive. If you don't, it's negative. Question? How are the proteins or what? How are the Rh formed? I would imagine there is instructions on the DNA that when you're creating the red blood cells, this is just part of the process that are included with it. So I guess that would mean what causes the interactions is what I'm meaning. My apologies, that probably should have been more clear for you. Yeah, because again, it's all about the interactions between, and it's caused by the antigens and the antibodies interacting. Yeah. Okay, so there are no corresponding antibodies in the plasma. So Rh antibodies are not present in your body normally, unless a person with Rh negative blood gets transfused with an Rh positive blood. So in other words, there's no reason for the body to make antibodies towards this antigen, this Rh antigen, unless it's present, right? And the first time this happens, it's not that big of a deal because the body just responds and takes care of it and life is good. It's the second time that there's problems. And the bell's going to ring, so please give me just like 30 seconds. How this comes into play. Here's a mother and a father who have opposite Rh factors. One's positive, one's negative. They have a baby. The first time, let's say the baby is a positive, the mother is negative. The mother's cells, some of the baby's cells get out into the mother, and the body sees this and reacts. Oh, let's make antibodies. Let's fight these positive things and get it out. That baby's fine. It's the second time that now the antibodies stay in the mother and they're ready. If they see another positive something, they'll attack. Well, now the mother gets pregnant again with, again, a positive blood-typed fetus, and now the body starts attacking. Because the antibodies are already ready, the response is already going, let's take care of it, and it can literally kill the fetus. Now, they have drugs that you can take now that will stop this. But back in the day, this was a significant problem. Because again, if you don't know, and I didn't say to my wife the first time I met her, so are you positive or negative? Because like I can't get together with you if, and we didn't know, right? Who cares, right? You wouldn't have thought of this, but in the past, this would have been a problem. Does that make sense? Cool. The only piece we've not talked about on your study guide are the list of different problems with the cardiovascular system. They are in the textbook. I'll give you a brief piece. I was hoping to get to them today, but not enough time. Okay? And I'll answer questions individually. Come to me. Adios.